inheritance patterns. The most beautiful female of the universe had met the ugliest professor of medicine and said, we'll both get married so that we get the most intellectual daughter born to us. Then the professor is not that easily lured into that proposal because he knows a bit of this genetics. He said, if the ugliest daughter like me and the dumbest with the dumbest mind like you, if she is born, what shall we do? So since she is the dumbest Miss Universe, she said, what is there, we will kill and once more try for the next baby. So, but professor is not even lured by that because he knows what percentage in the pedigree chart will lead to what probability of transmission of genes. Suppose if it is a 1:3 probability, even 3 times you kill also the ugliest and dumbest, still you can't achieve. So let's go through the inheritance patterns, doctor. Very, very important. So once upon a time the matchmaker used to tell, this guy is born with Saturn in the first house and this girl is having Jupiter in the third house. They will do well because ultimately they both don't leave the house in spite of fighting. Future may come, people may compare the pedigree, already they are doing that, of a few generations here and there, and one computer software program will do what is the future baby is going to become. Will he get in seat in MD or not? So there is that's what it will give. Probably that time somebody may say, I got second rank in entrance. You got 1000th rank. What is the chance of our baby getting? Computer may say 1000 to the power of 2 or 2 to the power of 1000. Only these are the two possibilities which are seen. It may give a reply. So let's talk. How do you draw the pedigree chart? So once upon a time in AIMS and All India MD exam, they used to give the questions which are based on pedigree and identifying it represents which type of inheritance, autosomal recessive, dominant, sex-linked recessive, dominant or a mitochondrial inheritance. Luckily there are only 5 patterns to be remembered. Okay, doc. Male is square. Female is circle. They are colored when they get affected, unaffected they are uncolored. If they die, you write an oblique into them. If he is an obligate carrier, then you put a central dot inside it. For example, somebody is having autosomal recessive disease expressed. When is it possible? Only when recessive allele from both the parents come, then only autosomal recessive will come in the next baby, next generation. Then what those parents are then called as obligate carriers. Unless they are definitely carrying at least each of them one recessive gene, the baby cannot show recessive disease. So such obligate carriers are given by putting one dot in the center. Marriage or mating. Marriage is after all a legal formality. Mating is the ultimate required formality. So then you put one single bar. But if maternal uncle's daughter, if mating is done, then you put double bar, consanguinity, double bar, right? Then proband. Proband means the guy in whom the first time disease is being discovered is called proband. You put one arrow pointing to the proband. This is what you need to be basically remembering. In pedigree, the generations, first, second, third, fourth are all Roman numericals, and individuals within a given generation are marked by Arabic numerals. Now doctor, let us start the game with autosomal recessive pattern of inheritance, the first pattern of inheritance. Typically, If a condition is expressed only if both the copies of the abnormal alleles are there, you call autosomal recessive. That's what our biology teacher taught us while teaching Mendelian inheritance pattern in our 8th class. Which kind of disorders are generally autosomal recessive, doctor? Rate limiting enzymes in biochemical reactions are there, no? Enzyme deficiencies are generally autosomal recessive. Favorite MCQ of the examiner, which you need to remember. Then in autosomal recessive, there is a concept called margin of safety. What is meant by margin of safety? Heterozygotes will express 50% of the enzyme activity because the normal allele will still produce 50% of enzyme properly. Abnormal allele did not contribute. Ultimately, if you take 100 micrograms enzyme expected of, let us say, carbonic anhydrase, in a guy with a deficiency, he will have 50% of it, right? But still that 50% will enable him to live normally. So the physiological function ultimately is not compromised. This is called the margin of safety effect, which is applicable in autosomal recessive disorders, is what need to be remembered. Then for anybody to show phenotypically the expression of an autosomal recessive disorder, important criteria is both the parents must be obligate carriers. One abnormal allele of one parent, other also should contain at least one abnormal allele out of the 2 alleles of a given gene. Then only the baby will have homozygous state for that autosomal recessive gene. Then only disease get expressed. Condition number 2. Consanguineous matings. The likelihood of both papa and mama carrying abnormal gene are greater. Why? When do you call consanguinity? Daddy's daddy and mummy's mummy are both brother and sister. Daddy's daddy and mummy's mummy are both brother and sister. That's how daddy and mummy, otherwise daddy is thought to be a useless fellow in the world. Nobody is ready to give a daughter, their daughter. Ultimately only his daddy's sister said, all right, my daughter also failed 10th class. Your son also is unable to pass 3rd class. Let both of them get married. At least our grandchildren will become cardiologists. So they both got ultimately married, right? So to marry once upon a time, they are called good older days. In those days, there is no need of degree. There is no need of post-graduation. There is no need of job also. The job itself is to get married. You turned 20 and girl, your mama's daughter has turned 18 means marrying her itself is an employment in the mama's house. He will have a garden, he will have everything available, right? So you can clean nicely. So if the mama's financial state breaks down, then the mama's second daughter also he will give you married, get you married and he will die. So they were all wonderful older days. Nowadays, are you MD? If you are MD. Are you clinical MD? No clinical MD. Are you DM cardiology? Okay. How is your practice? Where is your credit card? So many things are checked. Ultimately, sperm count is asked. Is it good or not? Testosterone levels are fine or not? How is the genotypic possibility of rarer alleles? Everything matching. Okay, we will consider. Then astrologer is called to check the planetary position. So, consanguinity, autosomal recessive disorders. Then some tribal areas. For example, you are living in Andaman Nicobar Islands. You are a part of the tribe. What is the criteria to get married? If you are able to kill the other tribe strong man, it is enough that you will protect the girl in future. So you have a chance of getting married. Their inbreeding is very high. There also autosomal recessive disorders are very, very high in frequency. If you do pedigree, what will happen, doctor? Pedigree. Typically in autosomal recessive, the disease phenotype is expressed by the siblings, but not by the parents or offsprings. What is the reason for that? What is the reason for that? Because parents are heterozygous, the expressing child is homozygous. That's the reason parents do not show any disease manifestations. Since it is autosomal, males and females equally are affected. Recurrence risk is how much for each sibling? One of the favorite questions of the examiner. 25%. This 25% autosomal recessive recurrence rate. Many times asked in exam. How it became 25%? Just you need to put that relaxedly and check how it became 1 out of 4. So, consanguinity. These are the things in the pedigree. So, this is an example of a classical pedigree. How will you recognize it is autosomal recessive? Consanguinity is one important clue for you to recognize. And if you take any affected guy, you take here or here or here or here affected guy. How are the parents of the affected guy? They are not affected. Is what you have to basically appreciate. Now, doctor, what are the various recessive pattern inheritance conditions is my next question to all of you. All the enzyme deficiency disorders, Tay-Sachs, Gotcher, Niemann-Pick, Hurler, Von Guyk, POMS, they're all the enzyme deficiency disorders which are all considered to be recessive pattern of pedigree. Similarly, Corey, McCardle, galactosemia, alkaptonuria, they're all autosomal recessive in nature. Then what is autosomal dominant nature? Even if one allele is abnormal, then there is a heterozygous state which will prevail and that lead to development of the disease manifestations. Now, doctor, what are the 4 possibilities in case of autosomal dominant type of inheritance? The first possibility is called haploinsufficiency. What is the meaning of it? Typically, out of the 2 genes, one is wild type, other is diseased allele, right? Even there is 50% normal activity also, basically autosomal recessive or dominant, abnormal gene is abnormal. What's the big deal? Abnormal gene is abnormal only. But why a abnormal gene leading to abnormality in autosomal recessive is not leading to problem, but in autosomal dominant why it is leading to problem? Because in these people, the haploinsufficiency is there. That is, in the autosomal recessive 50% enzyme is there also, still person is able to survive. In this, suppose if the 50% is not enough, for example, structural proteins, collagen, Ehlers-Danlos, Marfan, all these conditions, 50% collagen only is good, other 50% is abnormal. Can you have half the face good and other half abnormal? You can't have, no. So, the margin of safety will not help to prevent the normalcy, abnormalcy. So, even if 50% is good also, still other 50% will lead to the development of disease manifestation. That's how a heterozygote in autosomal dominant will lead to development of disease manifestation, is what we need to remember. That's called haploinsufficiency, where margin of safety doesn't work. Then second thing, suppose if the abnormal protein which is being produced is interfering with the functioning of the normal 50%, then also disease will manifest. That's the reason heterozygotes, so 50% normal is produced, the 50% abnormal is disturbing the normal 50%. That's called dominant negative effect is the nature. Then the next important situation is the abnormal protein is produced 50% only. But that abnormal protein has enhanced function. I mean enhanced malfunction. That is also leading to abnormality and cytotoxic effect in autosomal dominant. Then the next situation is, so these are the various mechanisms. Ultimately, bottom line is what? Heterozygous state also, autosomal dominant will be clinically expressing is what need to be remembered. Now, how will be the pedigree? What are the classical features in pedigree is a very, very important question. The phenotype of the disease, it appears in all generations. Grandpa had chorea, daddy had chorea, I too have chorea, because we are all not from Korea, but from Huntington's chorea. That's basically all generations. Autosomal recessive is not like that. If somebody had a disease, means he is neither of the parents should not have disease, will not have disease. So that's the reason whenever you draw a pedigree, how will it look like? The father of the nation is having problem, and the generation 2, the generation 3, the generation 4, so the proband is also having disease. Ultimately we detected the disease in this guy. That's the reason we are pointing towards it, proband. So that's the reason, doctor, it appears in all generations in the pedigree. Whenever a pedigree is given in the exam, immediately when you see multiple generations are showing, means you should call it as autosomal dominant, no second thought. Anywhere double line, consanguinity is there, means autosomal recessive, no second thought. No more analysis in the exam hall. Then, male and female it is equal occurrence, but few autosomal dominant conditions. Suppose if they are involving testis, they won't be seen in female. If they are involving ovary, they don't be seen in male, no. Such scenario where only one particular sex is involved, because that particular abnormality can only happen in that sex only, then you call sex limitedness will be there in autosomal dominant, right? Then what is the risk of transmission? 25% of autosomal recessive have now become 50% when it is autosomal dominant. Then is what you have to ultimately remember. Now, doctor, what are the various dominant heterozygous individuals? Why will they show the disease expression? We said haploinsufficiency. What do you mean by haploinsufficiency? 50% normal, 50% abnormal, but that 50% normal is not sufficient for the normal living. Alpha thalassemia trait and beta thalassemia trait are such examples. So 50% in them heterozygous state they are normal, but still the remaining 50% will lead to disease manifestations. Dominant negative effect. If you take osteogenesis imperfecta, or if you take Marfan's syndrome, the 50% abnormal protein will interfere with the 50% normal protein in a heterozygote. That's the reason ultimately disease will express. Then you have cytotoxic effect because of the dysregulated protein, we said, no. In case of Huntington's disease, what will happen? The trinucleotide repeats will lead to abnormal protein, Huntington's protein, and that typically has a cytotoxic effect of leading to degeneration of the nervous tissue. Hence, Huntington's chorea has become ultimately a neurodegenerative disorder, is what you have to ultimately remember. Then, so in autosomal dominant disorders, there is a phenomenon called as anticipation. What is meant by genetic anticipation, which is classical of autosomal dominant disorder. If you take a pedigree, grandpa had the first time chorea at the age of 62. His son, who is currently the top cardiologist, who is the associate professor of cardiologist, had at 43. And the grandson, who is still in the medicine MBBS final year, already is having chorea while shaking the hand with his fiancée. His fiancée is thinking the reason is something else. So, As the generations pass, early the onset, more severe the onset, becomes the Huntington's chorea. Now, doctor, X-linked recessive disorder and inheritance pattern, let us talk about it. Simple, the basic rule is, no daddy can give to his son. He can give anything to his son, but not what? He can't give a X-linked recessive disorder to his son. Even the poorest daddy in the world may not be able to give anything to his daughter, but still what he can give? X-linked recessive disorder. So, dad to daughter, but never dad to son is a feature which you see in X-linked recessive disorder. Then, if there is a heterozygous mother, right? So, what is the chance that the son of her can get affected? From a heterozygous mother, they have only 50% chance of being affected. That 50% doesn't happen from the mother. It is from the mother's husband, the father. Because mothers, can they be able to transmit anything? Will they transmit anything to the son? Yes, they transmit X chromosome. No. If that X chromosome comes from the heterozygous mother, from mother only it will come, that 50% chance. But 1 out of her 2 X chromosomes is going to come. If the good X chromosome of the mother comes, he is saved. If the bad one comes, he is getting affected. That's the reason heterozygous mothers will give 50% chance the sons to get affected, is what need to be remembered. So the incidence is higher in males, but not impossible in females. Why? If the mother is heterozygous for that X-linked recessive and father is already having that abnormality in the X, 100% chances father can give that X to the daughter, and 50% chance the mother can give to the daughter. So when daughter gets 2 abnormal things, then she can get affected. But son is not like that. So more common in males. Generally when daughter get affected, then she will only receive X chromosome abnormally from the dad. Assuming mother is not having any problem, the female heterozygotes are more likely to exist, and whenever they exist, they are unaffected carriers, is what need to be appreciated. Then sometimes what can happen is, there can be some new mutations. See, it is X chromosome, Y chromosome. X chromosome will decide color blindness, for example, or hemophilia. If it is a girl, what will happen if there is suddenly some radiation and a mutation in one X chromosome? The other X chromosome, which is not affected by radiation, which is a sporadic mutation, will prevent color blindness from coming. But in boy, he has one X, one Y. Suddenly radiation happened and one of his X got that color blindness gene on. Then there is no Y available to protect him. So, sporadic mutations can lead to the expression of X-linked recessive disorder to phenotypically express in the males. That's the reason we can have congenital color blindness, acquired form of color blindness also which can occur. So that's what we need to appreciate. So what are the classical X-linked disorders frequently asked in the exam, doctor, which you should not basically forget? Hunter syndrome, Fabry syndrome, which is due to alpha galactosidase deficiency, G6PD deficiency. Lot of times this was asked in the exam. Classical hemophilia, Lesch-Nyhan syndrome, Duchenne's muscular dystrophy. These are all the examples of the X-linked disorders, is what need to be basically appreciated. Now what are X-linked dominant? Do they exist? Yes, sir. Very few conditions are X-linked and dominant. Vitamin D resistant rickets, Rett syndrome, you remember in psychiatry. Then Alport syndrome, most of the cases, and incontinentia pigmenti and Charcot-Marie-Tooth syndrome, especially vitamin D resistant rickets, is one of the very high yield fact frequently tested by the examiner. Now coming to the pedigree chart of a X-linked dominant, how will it look like? X-linked dominant means even if one X is there, disease will appear. So all daughters of the affected men are affected. You agree with that? Right? But never their sons. Because affected man can never give his abnormal X to the son ever, even if he loves his son a lot. Right? So all daughters of the affected men are affected, but never their sons. When you listen this parable, what will you answer in exam? X-linked dominant. Recurrence risk is 50% for both male and female offsprings if the female is affected. If the female daughter is affected, then the recurrence risk is 50%. Then lot of times X-linked dominant disorders, there will be a lethality. In utero only they will die. They really don't grow old enough that they marry, they procreate, they produce, and then they donate that X-linked dominant. If you take vitamin D resistant rickets like scenarios, so prenatal lethality is characteristically a feature which you see in the case of the X-linked dominant conditions is what you need to basically appreciate. Now doctor, what is meant by incompletely dominant disorder? Incompletely dominant means if there is a heterozygous genotype in the case of autosomal recessive, how will he be phenotypically? Normal. Heterozygous autosomal dominant, how will he be phenotypically? Abnormal. For example, heterozygous autosomal recessive for idiotic nature is there, doctor. Then still he get 100 out of 100 marks in exam. Assuming exam is a good gold standard, good exam without no key mistakes. Mistakes in the key. If autosomal dominant heterozygous state is there for idiotic gene, he gets 0 marks. But incomplete dominance means if at all he has got a heterozygous state of this, he will get 50 marks. Not completely good like 100% autosomal recessive heterozygote, nor 0% bad like autosomal dominant, but in the middle of the two. Then you call incompletely dominant with an intermediate severity. One such example is sickle cell anemia, where normal allele is incompletely dominant in the heterozygotes. Suppose sickle cell trait is there. He will not show complete sickles inside his peripheral smear, nor complete biconcave disc, but 50% of the slide will show intermediate severity, or some precipitating condition is there, then only sickling will appear. So such things are seen in sickle cell trait. Commonly asked question, what is sickle cell anemia type of inheritance pattern? It is not autosomal recessive, autosomal dominant or anything, but incompletely dominant is what we have to remember. Now comes the mitochondrial disorders. We have a circular plasmid of mitochondrial chromosome which has got about 37 genes. They encode for RNA, tRNAs and rRNAs, and also for the oxidative phosphorylation mediating proteins inside the mitochondria. They are all decided by the mitochondrial chromosome. Mitochondria are there in cytoplasm. Cytoplasm is contributed by the ovum. Nucleus is contributed by the sperm. So, since ovum is the one who contributes the cytoplasm in which mitochondria is there, all abnormal genes come from the ovum, which is from the mother side. Which is from the mother side. Is maternally transmitted disorders will be the mitochondrial genetic disorders, is what need to be remembered. In this context, we need to know a term called as heteroplasmy. What is the meaning of this? Typically, in mitochondrial disorders, the affected cells usually have a mixture of mitochondria. The mutant mitochondrial DNA and also wild type of a mitochondrial DNA, both the things will be there, which is called as heteroplasmy, is what need to be remembered. So, why basically this heteroplasmy occur, doctor? If you take nuclear DNA, whenever the cell division is occurring, it is strictly segregated, right? Segregated into a haploid state from a diploid state. Mitochondrial chromosomes are not that strictly segregated like the nuclear chromosomes. That's the reason ultimately when abnormality of mitochondrial, the mitochondrial DNA inheritance comes, good mitochondrial chromosomes, bad mitochondrial chromosomes, both the things will be there because of an improper segregation, which is the nature of the mitochondrial DNA, unlike the strict segregation which is seen in the case of nuclear chromosomes, is what you have to basically appreciate. That's the reason what will happen in the case of the mitochondrial disorders, doctor? They don't follow the mathematical rules. The mathematical rule is, if a particular gene is there in 10 out of 100 in the generation 1, according to this probability law, this many number of abnormal genes should appear in the generation 2, this many in the generation 3, depending upon the mating pattern. The probability will be there, no, for the way the gene get transmitted down the generations in the population. That doesn't happen with the mitochondrial abnormal chromosomes. Because of that, we have what is called variable penetrance. In the generation 1, if 10 is there, according to the mathematical calculation, if you are expecting 100 in the generation 2, because of this much of mating pattern, instead of 100, you may only find 5 abnormal mitochondrial chromosomes only, because of this problem of issue of heteroplasmy, where all mitochondrial DNAs will not be abnormal. Some will be good, some will be bad because of an improper segregation, which is not that strict like the nuclear chromosomes, is what need to be remembered. Bottom line, heteroplasmy, mitochondrial DNA. That's what you need to understand. So, let's go through some of the important concepts in the genetics, doctor. What is meant by genetic heterogeneity? It means when different genotypes can lead to the similar phenotypes of the disease conditions, you call it as heterogeneity, genetic heterogeneity. A school teacher's son, police officer's son, and a bank robber's son, all of them become classmates in the medical school. Somebody who studied in California and the other studied in downtown in a small government school. So genotypes can be different, but ultimately phenotype becomes the same, then it's called heterogeneity. There are two types of heterogeneity: allelic heterogeneity and locus heterogeneity. So what is the meaning of it? When different alleles of the same gene produce clinically similar condition, you call it as allelic heterogeneity. So, because of this allelic heterogeneity, if you take a given family, what will happen in them? Though they have a genetic variability, still all of the members of the family express the similar type of phenotypic expression of the disease, which is explained by the allelic heterogeneity, because different alleles of the same gene produce clinically similar conditions. Then what is meant by locus heterogeneity? Typically, when mutations of more than one gene or locus can produce similar disease state, you call it as locus heterogeneity. For example, if you take Ehlers-Danlos syndrome, it can be due to mutation of nearly 10 different loci. Any mutation can lead to same Ehlers-Danlos ultimately, right? When the loci change, automatically the genetic allele position also will change. So, different loci are affected, ultimately Ehlers-Danlos is appearing. Some loci follow X-linked recessive pattern, some will follow autosomal recessive pattern, some loci which are abnormal will lead to X-linked dominant pattern. Ultimately, when you see the same Ehlers-Danlos patient, somebody's father also had Ehlers-Danlos, somebody's father has also Ehlers-Danlos, means which pattern it is? Autosomal dominant pattern. Somebody's parental level may nobody had, but he had, means it is autosomal recessive, right? So that is called locus heterogeneity, is what you need to understand. Variable expression and pleiotropy, what is the meaning of it? From one person to the other, the amount of severity of a particular genetic disorder will be varying. Okay, doctor? So, when I got second rank in MD, I was happily planning to join general medicine. But my first ranker is very having sweat in the palms. I said, why you are having sweat in the palms? No, I am waiting for the result in PGH Chandigarh. I asked, PGH Chandigarh, even Nizam's Institute of Medical Sciences is as good as any other top institute. Why you want to go there? No. I am waiting for my pharmacology MD seat there, he said. Hurry. We both got more or less a similar rank, but like a moron, I am taking MD general medicine. And like a top level scientist with a high level IQ, which is unmatchable, you are planning for pharmacology. So that's called as variable expression, nature and severity of phenotype ultimate, but that guy is now very top guy in the pharmacological circles, almost at the level of the drug molecule discovery level. Of course, at the end of MD, I am also discovering gun-like molecules among my students every day. So, variable expression. Then variable expressivity and penetrance, variable penetrance. Once more, these two are very characteristic of autosomal dominant disorders, is what you should not forget. Pleiotropy means what? Allele is the same, which is mutant. For example, chromosome number 3 is abnormal in two guys, but one had a cancer of the larynx, other had a cancer of the prostate. So, the phenotypic effect is different, then you call it as pleiotropy. Then what is meant by genomic imprinting? One of the favorite topics of the examiner. It's very simple. You have an abnormal gene coming from, let us say, chromosome number 15. There are two chromosome number 15s we get. One from Amma, other from Papa. From whom do we get that abnormal gene will decide which disease we get. So, if you get it, the chromosome, abnormal gene in the chromosome from the mother, then if that gene is shut off, then you call maternally imprinted. That means it won't express the disease manifestation if it comes from the mother. If the gene is silenced, that abnormal gene is being silenced when inherited from the father, you call paternally imprinted, is what you need to remember. And why will this occur? How will the gene know, I came from this fellow's mother and that's the reason I need to get silenced? Is it the adjacent gene will tell it? Or does it get a SMS from the God that, hey, your mother, it is the mother who brought you, not the father. So you should not silent, you must fully express. Will the God will send SMS? It is considered to be a epigenetic phenomena. That is, it is not because of any difference in the structure of that abnormal gene, whether it comes from father or mother, but it is the methylation reaction which will occur of some of the parts of the chromatin area at the time of the gametogenesis, which will make to get silented, and that methylation will occur only if it is coming from mother or whether it is coming from only father. So imprinting is a epigenetic phenomena mediated by the methylation of the two buzzwords, which you should not basically forget, doctor. Then comes genetic anticipation, which we have already discussed. Increasing severity of the phenotype and decreasing age of the onset as the gene is passing down the generations. So which conditions show genetic anticipation, doctor? Huntington's disease, Fragile X syndrome, wherever trinucleotide repeat mistakes are happening at the time of the DNA replication, that lead to development of the increasing mistakes as the generation is passing down. Hence, early expression and more severity of the disease is what need to be remembered. Then, what is meant by mosaicism? Some cells in the body are genetically different from the other. You call mosaicism. So, you have two kinds of mosaicism. Somatic mosaicism and germline mosaicism. What is meant by somatic? Typically, there is a mutation in a gene in a non-germline. There is a somatic cell in the early part of the development is basically called somatic mosaicism, so that ultimately the descendant is genetically distinct from the other. Germline means the parent's gonadal makeup. There is a mutation. That's the reason gametes will be able to transmit it. Only gametes will show that mutation. Somatic cells, if you pick out in the parent, it is not showing any abnormality. Only that mutation is seen only in the gamete is what need to be remembered. If you take most of the females and their genotype, technically speaking, they are all mosaic. Why? Because they have two X chromosomes, for the namesake two, but one X chromosome sleeps throughout life. It is inactivated. What is that hypothesis called? Lyon's hypothesis says that there is an inactivation of one of the X chromosomes. With only one X chromosome, they are very powerful. Our pathology prof used to say, if both X chromosomes happen to function, men should fight for freedom. So he used to say. So Lyon's hypothesis. But which of the two X chromosomes will undergo inactivation? Is there any definite mathematical rule for that? No, sir. It is very random X inactivation which will occur. So that is the reason what will happen. Even if there is any X-linked dominant abnormal gene is there on one of the X chromosomes, because of this lionization, if that particular abnormal gene, even though it is X-linked dominant, is a part of a inactivated X chromosome, then the disease doesn't occur. That is the reason X-linked dominant disorders may exist in lot of people, but due to the inactivation of the X chromosome, they won't be seen that frequently, because that X-linked dominant, even though it want to show its abnormal nature, it may be part of that inactivated X chromosome according to Lyon. That's the reason frequency of X-linked dominant will come down is what need to be remembered. If you take Turner's, classical example of mosaicism, up to 25% of the patients of Turner's, they show a mosaic karyotype, where only some types of cells only will show 45XO. Other type of cells, if you take from other locations, they may show 45XX. So that is a classical example of a Mosaicism is what need to be remembered. Then another important concept you need to know is called uniparental disomy, which we need to be very clear. I think all of our online classmates are disappointed today due to a pathetic transmission. I'm quite sure tomorrow we will try to set it all right to make the day shine better. So, but anyway, all this video we will try to push it out into the video library so that it should not be a problem for you to review. So, uniparental disomy. Typically, if there is any recessive allele, if it comes, say there are two recessive alleles, I mean a heterozygote means what? One recessive allele from one parent, another normal allele from the other parent. Suppose in some people, if both the abnormal recessive alleles happen to come from a single parent, only from one chromosome only is carrying both the recessive alleles, and the other chromosome, though it is normal, it is not having an abnormal allele, then the disease become expressed. Then you call it as uniparental disomy, where for the outside karyotyping it looks like a heterozygous state, but internally if you look, only one chromosome which came from one of the parents is carrying both the abnormal recessive alleles, leading to the disease expression, which is called uniparental disomy, is what you have to ultimately remember. So that is in short the story, doctor.